My name is Makala. I'm a PhD student at UNC Chapel Hill, and today I'll be talking to you about gut metabolomic changes during pregnancy, revealing the importance of gastrointestinal region in sample collection. This project was highly exploratory, so I'll be telling you equally about rationale, methods, and some results. So I wanted to start with just talking about the gut in general. I wouldn't be doing it any justice if I didn't mention a lot of its functional complexity. The microbiome is one of its more important characteristics, in my opinion. And the microbiome is defined as the collective genetic material of the microbiota, and the microbiota being bacteria, which makes up most of the gut microbiome, viruses, fungi, archaea, protists, algae, every microbe you can think of. And the microbiome is also involved in literally every other process in the gut, such as gut metabolism, which has both host and microbial contributions. All of this hinges on digestion and nutrient processing. And I think one of the most underrated aspects of this is how everything that is absorbed into the venous flow from the gut that's not directly used by the gut cells has to pass through the liver first before it can go into systemic circulation. Additionally, another really fun fact, I think, is that the gut has 70% of the immune system in it. It has a bunch of lymph nodes. It has these things called Peyer's patches, which are like lymph nodes that are embedded in the tissue of the gut, and they just survey the microbes and make sure they're not trying to hurt us. And then there are immune cells just within the tissue that are also surveying, but they're also supporting the enteric nervous system, which you may have been heard being referred to as the second brain. It is part of the peripheral nervous system, and if you sever its connection with the brain, it can actually still function on its own. So your gut will still contract, you'll still absorb nutrients and all of that. And I think the most interesting part of gut-brain communication is that 90% of that communication is actually the gut talking to the brain. And I think all of these complexities and all of the things that the gut does is why the gut microbiome in particular has really immense diagnostic and therapeutic power. Pretty much any disease you want to study will probably have some correlative changes in the gut microbiome, but the real challenge is determining if it's a response or if it's a cause. And so what I really liked about this webinar is that we're here to appreciate a more functional analysis of the gut microbiome that steps a little bit away from the paradigm of sequence and publish. The current workflow for studying the gut microbiome is really doing sequencing of the bacteria through 16S or the fungi through ITS in feces in your disease of interest. But I just want to quickly show you that there are so many things that can impact the composition of your microbiome. And that makes it really interesting to study, but it's also really fickle. So time of day can impact the composition of your microbiome. Your diet, obviously, can impact the composition of your microbiome. Where you live geographically can impact the composition of your microbiome. And your genetics can impact the composition of your microbiome. And that's not even it. Your gender, if you're pregnant, the way you were born, if you're on any medication, your environment, your age, if you have anxiety, all of these things can impact the composition of your microbiome. And it can be really overwhelming and difficult. And I'm not saying not to sequence, disclaimer, but I am saying that if you do, a metabolomics analysis would be a great complement for that. If you look at this figure here, this was done with the Human Microbiome Project data. And if you look at this top graph here, every bar represents a different subject. And so you can see that it's very jagged. There are different proportions of colors, and this represents the high variability of just fecal microbiome composition. But then if we look down here at metabolism, it's very stable across all the subjects. And so then using gut metabolomics, what can that even tell us? It can be difficult to parse out what's happening, but there's a lot that we know. Gut metabolomics can tell us either host or microbial metabolites that they each make on their own. They're really popular microbial metabolites that we already know microbes make on their own, like short chain fatty acids, certain polyamines, certain vitamins. And then there are popular host metabolites that we know the host makes on their own, like primary bile acids, primary conjugated bile acids, specific hormones and neurotransmitters. But then gut metabolomics can also tell us about interactions, metabolites that the host and microbe work together to make. And then it can also tell us about metabolites that the host can cause microbes to make, or microbes can cause the host to make. And so taking the full contents of the gut doesn't necessarily allow us to separate like the direct and indirect host and microbial metabolic response, but there is a lot that we know about how microbes and the host respond to certain base materials and what they'll make. And so that led to our study that was recently published. This was from my work at the EPA. Our goal was to characterize the difference in the gut metabolome in pregnant mice stratified by region. And then because we were using pregnant mice, we wanted to understand the influence of pregnancy on the fecal metabolome. So we also collected feces before these mice were pregnant. And so you're probably wondering, why did I choose pregnancy? Well, in my eyes, it was a really good place to start because we already know that pregnancy is a very, very metabolically challenging time. The mother has to shift her metabolism to support her and the offspring. We already know that in early pregnancy, the metabolism is focused on nutrient storage. And we know that in late pregnancy, the metabolism is focused on nutrient availability, accessibility, mobility, getting things to the fetus. And the liver is really the main hub of these metabolic changes. We also know that the gut microbiome changes a lot during pregnancy, and it's most drastic when you compare before pregnancy to the end of pregnancy. And we know that certain metabolites have been linked to this already that impact the immune system. So I thought this would be a great system to look at the gut metabolism in. And just a reminder, we are looking at the mouse GI tract, so it's not a human, but the overall anatomy, function, they have the same cells we do, their tissue is organized the same, all of that is shared. But the main difference is that in humans, there's a lot of microbial activity and fermentation in your colon. And in mice, the same activity is happening in their cecum rather than in their colon. And so we had to decide, how are we going to collect our samples? And as far as collection and prep before mass spec goes, it was really straightforward. As far as collecting the pre-pregnancy feces, mice just poop. And so when I was handling them before they were pregnant, I would just collect feces before it got into their cage. And during necropsy, I just would take each gut section and very gently push out the contents of the tissue into a tube. Everything was immediately frozen. And then later, we weighed the samples for normalization for metabolomics. And speaking of metabolomics, I had to figure out, okay, now I have these samples, like how am I going to analyze them? And I happened upon the biocrates MXP Quant 500 kit. And a lot of the things they had on their website I really liked, that I could use 10 microliters of sample, love that, working with mice. That there's a fully integrated workflow, and we didn't have to develop anything. I'm not a chemist. I didn't physically do the mass spec. So I love that too. And it seemed to me that, I mean, from the title, they cared about the microbiome and the metabolites that the microbes were making. And so I saw that there were a lot of microbial metabolites specifically that were included in the kit. And so I was very excited about that. And so what did we end up being able to pick up with the kit, just so everyone knows? We used mice. We were able to pick up all the small molecules except for two of the fatty acids. And we weren't able to pick up any of the lipids. It could either be because of our method, but it's most likely because we used mice and because we used the contents of the gut rather than like the tissue itself. And so what did we find? First, I'm going to talk about pregnancy. What did pregnancy do? I'm briefly going to pick out a few of my favorite changes. And if you're interested in others, you can read the paper. But one of the most interesting changes to me is that there were 11 amino acids that were altered by pregnancy, and literally all of them were increased by pregnancy in the feces. And Alice talked about this earlier, but that could mean that there is an excess and it's going to be excreted. It could mean there's an excess and it's going to be reabsorbed, but these were fecal pellets, so that's not likely. Either way, to me, this indicated that there was a likely upstream change in amino acid metabolism or mobilization, which really fits well with the idea of increased nutrient availability to the fetus in late pregnancy. Something else that is just of interest to the field are bile acids. So I'm going to briefly mention the bile acids that changed here. And before I talk about that, I feel like I need to talk about bile acid metabolism because it's a little bit complicated in the gut. But very briefly, cholesterol is converted in the liver to these primary bile acids, and then the liver adds either glycine or taurine to them. That is then stored in the gallbladder as bile salts and excreted into the duodenum, which is the beginning of the small intestine. It's mostly unchanged through the small intestines. And then by the end of the small intestines, most of that is reabsorbed and goes back into the liver and is recycled. But some of that gets past the small intestine and goes into the colon, where microbes can do their magic on it and they generate secondary bile acids, which is kind of unclear in the literature, like what happens next. But we know there are conjugated secondary bile acids, but no one really ever says like directly if microbes are making it or if it's secondary bile acids that are reabsorbed and then conjugated by the liver. But I would like to think both of these things could happen. And so what we found was that pregnancy specifically increased a secondary conjugated bile acid and decreased a primary bile acid from the liver and a couple more secondary bile acids. Another thing I was really interested in were the fatty acids. 80% of them were altered by pregnancy. And honestly, I expected that pregnancy would decrease a lot of the fatty acid availability because this time is a time of a lot of lipid utilization and mobilization. But we actually found that two of them were increased in the feces. And I'm thinking this might be because these fatty acids are associated with early pregnancy and early fetal development. So maybe it's more necessary early rather than late. And then region. What did we find with region? We found that gut region really matters when you're looking at metabolites in the gut. And the two regions I thought would be the most different were the small intestines and the cecum, mostly because of what they do and like just their microbial contribution. And that is what we saw And we see here that compared to the cecum, a lot of the metabolites that were decreased in the small intestine make sense. Trigonylene is a big microbial metabolite. You would expect that to be higher in the cecum. It has a lot more microbes. Same with secondary bile acid, the secondary bile acid here, and indole and hypoxanthine. These are also microbial metabolites. So we're sort of seeing what we kind of expect. We also see some changes that may be related to function. We're seeing that amino acids and amino acid related molecules are decreased in the small intestine compared to the cecum, which is probably related to amino acid uptake. And then we see that all of the fatty acids that changed are decreased in the small intestine compared to the cecum, which also makes sense functionally because fatty acid metabolism mostly occurs in the colon. Again, in comparing metabolites that were increased in the small intestine compared to the cecum, we see a lot of changes that were kind of expected, but we see some that I didn't necessarily, I couldn't predict, why is cysteine availability so high in the small intestine versus the cecum? That was the only amino acid that was higher there than it was in the cecum. We also see a few bile acids that are higher in the small intestine, which we expect. Primary bile acid Ca, primary conjugated bile acid GCA, and TC-DCA. Those are expected to be higher in the small intestine. But we also see 2 secondary conjugated bile acids that are higher in the small intestine than in the cecum, which makes me think that bile acids are being taken back up from the colon and reconjugated by the liver and recycled back into the small intestine. We of course compared small intestine to fecal. Again, there were some expected differences. Lower in the small intestine were microbial metabolites such as trigonylene, the secondary bile acid, indole, the fatty acids, and increased are the same biogenic amines as in the cecum. We see, again, those primary bile acids are increased in the small intestine compared to the feces. And then cecal and fecal comparison, I kind of didn't expect many changes, but there were a lot more there than I guess I expected. Metabolites that were decreased in the cecum compared to the feces were some biogenic amines and indole. This indicates that there's more microbial contribution in the colon than there is in the cecum. And then in the cecum, pretty much everything else was increased in the cecum compared to the feces. Cecum has much more microbial activity, more fermentation, metabolic processing than the feces, which I think explains a lot of these changes. And something that we did, which I really liked, was that we used this computational tool called Ingenuity Pathway Analysis, or IPA. And we mined the human metabolome database. We put in the levels of metabolites that we saw, and it gave us pathways and things that were related to the metabolites. So I like this because we were able to see a lot of expected differences between the regions. Like in the small intestine, you see some depolarization of neurons and cyclic AMP that's related to the enteric nervous system. In the cecum, you see that the metabolites are related to bacterial growth, which makes sense. Growth of organisms, growth of Yersinia pestis. In the fecal metabolome, you see that there's a lot of amino acid uptake, and fatty acid metabolism and synthesis, which is exactly what we expect. But there were also some really interesting things that we picked up, maybe some sort of novel nuances. Like in the small intestine, we see synthesis of DNA and nucleotides. Maybe that's related to turnover of the cells. In the cecum, we see um, some amino acid transports, and then in the feces, we see blood pressure, which maybe makes sense with like fluid and things. But very interesting things came up from that. So really what's like left now, something big is that we didn't look in the tissue metabolome and we didn't look in non-pregnant mice. So that would be a great study, establishing a complete gut metabolome regionally in mice. It would be great to explore how these different GI regions respond to which disease states and changes in pregnancy. And then testing regionality. We can identify different nutritional, chemical, environmental exposures or interventions that alter the gut metabolome. And we can look at where this happens. And that would be really interesting too for someone to do. And so I just like to say thank you to everyone. Thank you to the main people that really worked on the paper and the study, Dr. Colette Miller, Dr. Thomas Jackson, and Jermaine Ford, who ran the metabolomics. And if you have any questions, you want to read the paper, please reach out and contact me. And special thanks to the mice, always.